Okay, I got it. Okay. All right. Looks like everyone is muted, so looks like we're good to go there. All right. Oops. Missed one. There we go. Okay. All right. So I am very excited to tell you all today about my dissertation research, Bioinformatic Approaches for the Study of R loops. So first, a little bit of background on R loop biology. So R loops are three-stranded nucleic acid structures comprising an RNA-DNA hybrid and displaced single-strand DNA loop. They form naturally during transcription as nascent RNA leaves the polymerase and reanneals to the template strand. R loops are typically studied either in the context of their pathological consequences or in the context of their physiological roles. And so I'm going to jump right into talking about the pathological context of R loops because it is the most well-studied to date. So the pathological context is particularly interested in attempting to understand the balance between R loop formation and R loop resolution. So the factors that promote R loop formation include those such as negative supercoiling, also known as negative superhelicity, and G content. And when we're talking about G content, we're really talking about this right here, so-called RIS-RES regions. Now, RIS regions, also known as R loop initiation zones, are regions that contain a large number of G clusters. There's also a linker region of any sequence content. And there are also res regions, R loop elongation zones that contain high G content, but not necessarily G clusters. Okay. These RIS-RES regions are also known as R loop forming sequences, or RLFS, and they can be computationally predicted using specialized software. Okay, so those are the factors that can promote R loop formation. Now, there are also a variety of factors that are involved in either preventing R loop formation or in actively resolving R loops, including factors such as RNase H, which you see here highlighted RNH. And again, the pathological context of R loop biology is really concerned with the balance in between these factors that promote and prevent R loops. And then moreover, what happens when you lose some of these R loop resolving factors and then that balance is lost. And so in this circumstance, we get what's called a pathological R loop, also known as an unscheduled R loop. And because of how thermodynamically stable R loops are, these do not go away on their own. And so when you artificially knock down or knock out these R loop resolving factors, it can lead to a constitutive R loop, which has several pathological consequences. For example, the exposed single-strand DNA loop can be prone to mutagenesis. And if you have a dividing cell population, you could encounter transcription replication conflicts, so conflicts between the transcriptional machinery and the replisome. And this can lead to hyperpositive supercoiling at the interface, as well as fork stalling and, if unresolved, fork collapse and recombination. So again, the pathological perspective on R loops is really concerned with the idea that R loops are tightly controlled by this balance of R loop formation and R loop resolution, and that when that balance is disturbed, you can encounter mutagenesis, replication stress, and recombination. Okay. Now, as I mentioned, that is the most well-studied context for R loop biology thus far. However, as I've already alluded to, the pathological formation of R loops is typically promoted by these sort of artificial gene knockouts or knockdowns. It doesn't tell us as much about what R loops are doing under normal basal conditions. And so for that, we jump here to the physiological context of R loops. Now, this came about because of the pioneering work of my external committee member, Dr. Frederick Shedin. And in his 2012 study, he showed using a technique called R loop mapping, which I will get into a little bit later on, that R loops actually form genome-wide at unmethylated promoters. and moreover, that they can actually prevent promoter methylation by blocking the activity of DNMT3B1. And we see that here displayed. So in essence, what this showed is that R loops are not inherently pathological, and they can also play key physiological roles. Okay, in the decades since that study came out, there have been a variety of studies that have revealed numerous physiological roles for R loops. These include, again, promoting unmethylated DNA, and that happens by blocking the activity of DNMT3B1, but also by promoting the activity of GAD45A and TET, which can lead to active demethylation of the DNA. Additionally, R loops can promote antisense transcription, and there are a variety of mechanisms and contexts by which this can occur. Finally, R loops can also promote histone modifications, and here is a diagram showing a few of the ways in which they do that. Okay, so we've now seen that there is a physiological context for R loops and some of the roles that have been described thus far. But there are many more questions that remain unanswered today, such as what are the other types and classes of physiological R loops? What other roles and relationships do they participate in? And how do they change under differing biological conditions? And so these were some of the questions that I wanted to address during my dissertation, and in particular, trying to understand what are the other epigenomic roles and relationships of R loops. Okay, so the approach that I took involved R loop mapping and data mining. And very basically, this approach began by reprocessing, standardizing, and quality controlling R loop mapping data sets, moving on to a meta-analysis to study R loop classes, and then finally wrapping up by looking at the association of R loops with specific epigenetic elements. So I'm going to jump right in here and talk about the first step in that process. So just to get a little bit of context and background into R loop mapping, it was first developed by my external committee member, Dr. Shadin, in 2012. And the first R loop mapping technique described was called drip sequencing. Now, because this is still the most prolific R loop mapping technique in the literature, and many techniques are based upon it, I felt it would make sense for me to just briefly walk you all through the steps of drip sequencing. So first, genomic DNA is extracted and then fragmented with restriction enzyme digestion. Some of those fragments will contain R loops. The R loop containing fragments are immunoprecipitated with the S9.6 antibody. That's an antibody that is relatively specific to R loops. The fragments are washed and eluted and then sequenced. And just to make a brief note about what sets us apart from some of the other techniques we're going to be talking about later, the immunoprecipitation here is happening on genomic fragments that have already been extracted from the tissues, have been fragmented, have been treated with proteinase, and so we consider this to be an ex vivo approach of R loop mapping. Okay, so what does the data look like when it comes back from the sequencing core and gets processed? So to help illustrate, I'm showing here a screenshot of the genome browser, and we're looking around a region of chromosome 7, nestled in between two genes which seem to share a bidirectional promoter. Okay, so there are 4 samples in the study, and these are actually samples I processed myself as part of the work done in the second to last chapter of the dissertation. So these are the first 2 samples, and we can see here the coverage. And basically, this is a histogram that shows the number of sequencing reads that align at any given point in the genome. And you can think of this like the R loop signal. And from this, we can see very clearly that there is, in fact, an R loop right here. We also see peaks, which are the discrete and normalized form of that signal. Okay. And so whenever someone does an R loop mapping study, they typically make a claim about the number of R loops detected, and they're typically just counting the number of peaks that they called. Okay, So in this study, we had two conditions. These are all cells that overexpress wild type BRCA2. And we also have two samples that have mutant BRCA2. And so these are those samples here. And we can see very clearly that the R loop here is not only stronger, but it's also wider. And so that is really the power of drip sequencing and other R loop mapping assays, is they can tell us the differences in R loop formation that occur due to biological condition. Okay, so that's drip sequencing in a nutshell. 4 years later, Dr. Shadin's group published an update to drip sequencing called DRIP-C sequencing, which was strand specific and sequenced the RNA moiety in the R loop rather than the DNA moiety. 1 year later, Chen et al. Published a very different approach called R-CHIP, which uses catalytically dead RNase H1 in order to map R loops. And just as I kind of explained earlier, RNase H1 is an enzyme that can bind and degrade R loops. Catalytically dead RNase H1, however, can only bind R loops but not degrade them. So this is basically how R-CHIP works. You consider some region that has the propensity to form R loops in this cell line that overexpresses the dead RNase H1. So the dead RNase H1 will bind to those R loops, and then we can use chromatin immunoprecipitation to chip for that dead RNase H. And thus that allows us to map the locations of R loops. And as I'm just drawing this comparison again back to drip sequencing, we can see not only sensor used for mapping R loops different. I'm going to go mute that person real quick. If you could please turn off your microphones, everyone. Thanks. All right. Okay, got him. All right. Yeah, very basically, while drip sequencing is an ex vivo approach where the genomic DNA has already been extracted, with R-CHIP, the R loop sensor, the dead RNase H, is being applied in vivo. So it's being applied in the native chromatin environment of the cells. And thus we expect that these sort of approaches will produce somewhat different results to some extent from drip sequencing and the other S9.6 approaches. Okay. Oops. All right, so at present, there are a wide array of R loop mapping assays out there. Here's a mind map showing a few of them. Here we see the S9.6 based approaches, such as drip sequencing. We see the dRNH based approaches, such as R-CHIP, and we see other miscellaneous approaches as well. All right, and that is R loop mapping in a nutshell. So coming back to our approach here, we began by taking R loop mapping studies, manually curating them, and then using a data processing pipeline to standardize and reprocess those data, building a database of those data, performing meta-analyses. And the ultimate goal was we would be able to do all this and address some of those fundamental questions about R loop biology that I brought up earlier. However, there were a few outstanding questions remaining about the feasibility of this study. In particular, one issue was, of course, how do you manage the difference between R loop mapping assays and modalities? And just to refresh your memory, there were quite a few of them, and we expect at least dRNH and S9.6 to give us somewhat different results. And then two, how do you manage issues of data quality? And the reason that this got brought up was because of a study that I had the opportunity to be a part of that got published in NAR back in 2020. And in this study, I standardized and reprocessed about 100 drip sequencing data sets and came to realize that quite a few of them were of low quality, and even a few more actually didn't seem to map any R loops at all. So this is what I mean. So this is a view of a genome browser. It shows about a megabase span of the genome, and it's centered on the NEAT1 RNA gene. Now, NEAT1 is a gene that shows some of the highest and most consistent levels of R loop formation across studies. Now, we're looking here at one of our drip sequencing samples, and we see, as expected, it maps an R loop right there, and it seems to also map a few other R loops across this region of the genome. See, another sample from a different study also maps that R loop, and yet we see two other samples from two different studies in red here that not only seem to fail to map the NEAT1 R loop, but also don't seem to map any R loops across this entire region of the genome, thus suggesting very strongly that they are not actually mapping R loops. We weren't actually the only ones to notice this. Dr. Shadin and his recent EMBO commentary found very similar results. And so I'm just showing you a figure from that paper. So they looked, instead of at NEAT1, they looked at RPL13A, which is another gene that has very high and consistent levels of R loop formation. And so we see here several samples that map that R loop quite well. Here are the negative controls for those samples, so we don't expect any R loop mapping in these negative controls. And yet again, we see another two samples from two different studies where the positive sample did not map the R loop. And not only that, but it does not look very different from the negative control. And so that just really reinforces the idea that for this approach to be successful, it's going to need an extra step, and that step was going to have to be quality control. And that takes us right into the main study that I want to talk about today. This was published just a couple months ago in Nucleic Acids Research. It's called Quality Controlled R Loop Meta-Analysis Reveals the Characteristics of R Loop Consensus Regions. Okay, so in this study, I began by manually curating the R loop mapping data sets using sources such as Geo and PubMed. This is a screenshot of the Excel spreadsheet in which I curated the data, and there are about 810 entries in total. The next step was to use a custom-built data processing pipeline called RL Pipes in order to process the data uniformly. So RL Pipes can take pretty much any type of R loop mapping data, and it can perform quality control on the raw reads themselves, as well as alignment to the genome. And then it results in called peaks, coverage, and alignment statistics. Of note, RL Pipes, along with some of the other software that we developed during the study, is described in a recent bioRxiv preprint, which we put out there just a couple weeks back. I'll mention that again a little bit at the end. So one of the first things that I looked at was the number of peaks called per sample split by the R loop mapping assay used. And so we can see here some pretty stark differences. There are some assays that seem to map hundreds of R loops and some that seem to map hundreds of thousands of R loops. And so there's just tremendous inter-assay variability. And not only that, but even when you look within some assays, you can see very clearly there is a lot of variability occurring. This poses a few problems that I felt we would have to address, such as the variance within and between assays and modalities, which is really problematic because there is no ground truth. We don't know for sure where the real R loops are occurring. And thus it makes it very difficult to judge whether it should be hundreds of R loops or hundreds of thousands. And so to try to address that idea, I used R loop forming sequences as a sort of independent test of where R loops are being mapped and of which of these assays appears to map R loops more effectively than others. Now, the idea here is, again, R loop forming sequences show a propensity for R loop formation, and not all of them will have an R loop in them. And also, not all R loops will occur in an R loop forming sequence, but we expect enough of a statistical enrichment between the two that we can use them as a test of whether or not an R loop mapping sample is actually mapping R loops. And while I don't have time to tell you exactly how I did that and how I was able to show that it was both internally and externally valid, that is described in our NAR paper. And I'm instead just going to jump right into the process that I used. Okay. So first step was to analyze R loop forming sequences, following that up by building a machine learning based model. So that would allow us to classify our samples as either mapping or not mapping R loops. Then we took the high quality samples that passed through our model successfully, and we used them for our downstream analysis. Okay, just a couple of abbreviations to keep in mind throughout the rest of this talk. First is DRNH. This just stands for catalytically dead RNase H1. And this includes techniques such as R-CHP. And then S9.6, which refers to S9.6 based techniques such as drip sequencing. Okay, I want to also draw our attention to positive and negative. Positive means that the quality model, the machine learning model, returned a prediction of positive. It thinks that the sample does map R loops, whereas negative is the opposite. It thinks it does not map R loops. And just to see what that translates to in reality, I'm going to show you the genome browser view from earlier, and I'll overlay on top of this what the model predicted. So for these first two, the prediction was positive. For the second two, the prediction was negative. Okay. So let's jump right into that meta-analysis. So the first step, as I've already mentioned, was to apply the machine learning-based quality model. And as I've kind of already alluded to, we saw that there were differences, at least we predicted there would be differences between DRNH and S9.6 based techniques. So we split them into their own bins and analyzed them separately. So for DRNH, about 3/4 of the samples passed our filter, about 2/3 of the S9.6 passed the filter. So we took these high quality data sets and we used them to build consensus peaks, consensus R loop peaks. And I'm going to show you how that worked using a little illustration. So let's say that we're looking at a region of chromosome 7 here, and we've got 10 R loop mapping samples that all have peaks within this region. If we were to window the genome and we were to count up the number of peaks overlapping each window, it would probably look something like this. Now, these sort of mountains have different heights. So we can see the first one is 6 peaks, the second is 3 peaks, and the last one is just 1 peak tall. We wanted to make sure that when we called peaks on these mountains, that they really were robust and really represented true R loop biology. And so to help us do that, I set a quality cutoff of 15%. Basically, for any consensus peak to get called and to be valid, it had to be supported by at least 15% of the high quality samples. And so in this case, that means that we only have 2 consensus peaks from this analysis, one with a score of 6, representing the height of the mountain from which the peak was called, and one with a score of 3. Okay. All right, they've muted themselves. Okay, good. So then we took these DRNH and S9.6 consensus peaks, and we used them for our meta-analysis. Okay, so roughly 35,000 consensus peaks were found for each. And then when we overlapped them, I saw here that, very interesting, about 10,000 were shared between them, and that overlap was also quite significant. But it was really curious to see that even with that much overlap, there was also a lot that was different. Now, I'd already alluded to the idea that we expect these to map somewhat different populations of R loops, but this seemed like a lot more than I was anticipating. And again, we have set a very high bar for the quality of the samples we used here, as well as a high bar for what constitutes a consensus peak. So we're quite confident about these. These are not spurious, and they likely represent some facet of true differences in R loop biology that I wanted to explore further. And so I then asked the question, well, okay, what makes these R loops different from each other? And so the first thing that I looked at was the size of the consensus peaks. And I saw this is about the size for DRNH, and S9.6 was a little bit larger. And looking at the genome browser here, we can see some of the samples, and we're actually looking here just to orient us. We're looking at the COM2 gene, and that is one of the most mapped R loop locations in our study. Just to orient, we see here, this is the TSS and promoter. This is the gene body, and this is the transcription termination site, TTS, and poly A site. So this gene is going from right to left. Okay, Again, just to orient, I'm going to show a couple of our high quality R-loop mapping samples. So here we see one of our DRIP sequencing samples, one of our DRIP-C sequencing samples, and one of our S-DRIP samples. And then here we can see the consensus S9.6 signal and peaks from our consensus analysis. Okay, we can see that this shows R-loops being mapped at the beginning, middle, and end of the gene. If we jump over to DRNH, we see our R chip here. We see our R chip, and then we see map R. And then when we look at the consensus, we can see that it, unlike S9.6, appears to be mostly located more at the beginning of the gene. And so this was really interesting because it actually evoked a model that Dr. Shadin, my external committee member, proposed in a recent DNA repair review paper. And this is that model. Basically, it's the idea that there are at least two classes of R-loops. Class 1 R-loops, which are associated with promoter proximal pausing, and class 2 R-loops, which are associated with transcriptional elongation. And just to help understand what I mean when I'm talking about pausing and elongation, here is just a quick model. Basically, the idea is that transcription initiation involves the PIC, which attaches to the chromatin and transcribes for a little ways, maybe 60 nucleotides or a little bit longer, and then it pauses. And this involves the activity of NILF, and typically paused RNA polymerase has a serine 5 phosphorylation on the C terminal domain. Now, then the pausing is released partially through the activity of PTEF-B, and this leads to serine 2 phosphorylation and the resumes activity of the polymerase. So this is pause release. This activity of initiation, pausing, and pause release is a conserved feature of mammalian genomes, and certainly there are a very large proportion of human genes which show this same propensity. Okay, so that is pausing and pause release. So the idea is that these class 1 R-loops are associated with the pausing of the polymerase, and the little bit of nascent RNA it's already produced forms that R-loop. What else this model predicts is that RNase H1 under native chromatin conditions can access those class 1 R-loops. And so we would expect that those R-loops can be mapped by S9.6 and DRNH-based assays. Alternatively, with our class 2 R-loops, we expect that those are probably really just detectable by S9.6 alone. And so we wanted to ask next, do we see this pattern? And so I decided to do a metagene analysis. And basically what I'm going to show you is the aggregate or average DRNH and S9.6 signal across all genes in the genome collapsed and averaged together. And so we see here DRNH, mostly at the promoter proximal parts of the gene, and S9.6 at the beginning, middle, and end of the gene. So this seems to provide at least a little bit of preliminary support for this model. But I really want to go further. And so this is just a little bit simplified version of the same model. Basically, I want to test some other facets of this model, such as the idea that your class 1 R-loops should be shorter and your class 2 R-loops should be longer. And as I had already seen from our earlier results, that was in fact the case. So another aspect of the model that I wanted to address is the idea that because your class 1 R-loops are really just associated with promoters, and your class 2 R-loops have the whole gene body to work with, we would expect the class 1 R-loops to cover a much smaller proportion of the genome. And so in looking at the human genome, about 3,200 megabase in total, we zoom in here on the region that is covered by our R-loop consensus sites. And that covers about 139 megabases, or 4.32% of the genome. So in breaking that down by S9.6 and DRNH, we can just see right off the bat that most of that distance is actually covered by S9.6 consensus peaks. Now, just as a note, I have broken this down into S9.6 only and S9.6 shared. Now, S9.6 shared, those are S9.6 consensus peaks that overlap with at least one DRNH consensus peak. S9.6 only, those are consensus peaks that do not overlap with any DRNH consensus peaks. Okay, Looking at DRNH now, we can see that just side by side, it clearly covers a much smaller proportion of the total genome. Okay. So the next facet that I wanted to look at is the idea that these class 1 R-loops are associated with particular transcript features, such as the transcription start site and promoter proximal regions of the genes, whereas class 2 R-loops should be associated with gene body features. Okay, so in doing a transcript feature overlap analysis, we see here the transcription start site, 5' UTR, these are gene body features now, 5' UTR, exon, intron, 3' UTR, and transcription termination site, along with intergenic regions. Those are regions that don't overlap with any genic features. And so in answering our question now, coming back to the transcription start site / promoter regions, we see here that DRNH only consensus peaks are certainly more enriched within these sites. Whereas when we look at the gene body features, there's certainly more enrichment for S9.6 here. So again, that seems to support the idea of our class 1, class 2 model. Finally, I wanted to look at one other key facet of the class 1, class 2 model, which is the idea that your class 1 R-loops are associated with promoter proximal pausing. Okay, and while I don't have time to fully get into exactly how I was able to go about measuring this, that is fully described in the NAR paper, I'm just going to jump right into the salient findings here. So basically, we were looking at the genes overlapping, the R-loops that were more or less specific to DRNH or more or less abundant in DRNH. And the idea being that the DRNH abundant or DRNH high R-loops should be the ones that overlap with more paused genes. And so to measure that, we're using the pausing index. And so in looking at the results here, we see that in fact, yes, the DRNH abundant R-loops are in more paused genes. And not only do we see that in HEC293 cells, as you can see here, we also looked at another 11 cell lines and saw very similar results. So coming back to our model, that really does seem to suggest that yes, in fact, this model is, at least in the data we were able to process, this model is sound. Now, one caveat though, that is worth mentioning about this model is that it has a bit of a blind spot. And this is basically that blind spot. It kind of presupposes that when you overlap DRNH and S9.6, you should see something like this. DRNH should be something of a subset of where S9.6 is mapping R-loops. And yet that's not what we saw. We saw a very large number of DRNH only R-loops, and they cover a sizable proportion of the genome. So I really wanted to dig in and start to ask the question, well, okay, what then are these DRNH only R-loops? And so for the first way that I wanted to address that was just by looking at the size of the consensus peaks themselves. And we see here S9.6 and DRNH shared, roughly similar sizes, but DRNH only is much smaller and quite significantly so. Okay, so they are smaller. I also wanted to look and see if they are associated with genes that belong to particular gene programs. And so with S9.6 and DRNH shared, we can see that those are very strongly enriched within certain gene programs, but DRNH only was enriched within no gene programs whatsoever. And so they are not part of a gene program. Oops. I wanted to then look and see if, well, okay, maybe they were associated with particular transcriptomic features. And so looking at S9.6, we see those are mostly gene body features. DRNH shared, mostly transcription start site and transcription termination site. But with DRNH only, I noticed very interesting, a full third, in fact, more than a third of those consensus peaks are actually intergenic. They don't overlap with any transcript features. And what this kind of implied to me is that, well, potentially they are overlapping with cis regulatory elements. And so that's what I wanted to look at next. I looked at the enrichment of these consensus peaks within these cis regulatory elements. First, looking at enhancer promoters, no clear specificity for DRNH only, same with promoters. And then looking at distal enhancers, now I could see there was very clearly a specificity for DRNH only. Looking at in a little bit greater depth, this is a metaplot that's going to show the pileup of DRNH only, DRNH shared in S9.6 around distal enhancers. And we can see that DRNH only, very strong enrichment. The others are pretty much flat lines. And then if you look at the overlap of intergenic DRNH only peaks with distal enhancers, more than 2/3 of them are overlapped. So again, suggests very strongly that the DRNH only is going to these enhancers, and it also takes us very neatly into the very last part of the study and also this presentation, looking at the association of R loops with specific epigenetic elements. Okay, before I jump into this, I'm just going to briefly tell you a little bit about how enhancers work and really what they are. So, enhancer is a region of open chromatin with active histone marks and transcription factor binding sites. There are mediators and cofactors that help to establish a 3D interaction between the enhancer and a target gene promoter, and this leads to the target gene being turned on. I want to focus very briefly here on the enhancer element itself. These are very interesting. They're actually themselves actively transcribed into a species of long non-coding RNA known as enhancer RNA, or eRNA for short. Now, while the function of enhancer RNA remains something of a mystery today, there is compelling evidence to suggest that eRNA can, in fact, form R loops. And so I wanted to dig in a little bit further, use our DRNH-based samples, and try to understand, okay, can we characterize these eRNA R loops? And so this is the approach that I took. Using data from 2 cell lines, iPSCs and Cuttle1 cells that both have relatively well-defined enhancer profiles. Taking enhancers from the gene enhancer database, getting R loop mapping data from MapR samples, so it's a type of DRNH-based R loop mapping, and then getting Bro sequencing, which allows us to assess nascent RNA production. Okay. This allowed me to predict eRNA R loops, and then I used the data from the cell marker database, gene enhancer interaction database, and Hi-C sequencing data, which measures 3D interactions to characterize those eRNA R loops. Okay, so jumping right into the results, this is the data from, this is the MapR data from iPSCs showing the pileup around distal enhancers. And this is showing us 4 replicates here. These are all high quality replicates. They've all passed our quality model, just for clarification. And so this shows pretty clearly that we are finding R loops at distal enhancers within this cell line. Jumping over to Cuttle1, we can see with 6 replicates, almost all of them show very clear enrichment in distal enhancers as well. I wanted to look next at the levels of nascent RNA production at these enhancers. So basically what this shows us here is the level of eRNA transcription at the enhancers that either have R loops or do not have R loops. And as we would expect, the enhancers that do have R loops have significantly higher rates of eRNA production. And just to be clear, this is a log scale we're looking at here. So just judging by the significance, this is quite a big difference. And we also see a clear enrichment in Cuttle1 cells as well. So these are, in fact, eRNA R loops. Looking now at the genome browser at an example of one of these, we see that this is clearly a distal enhancer cluster right here based on the gene enhancer track. We also see the 3D interactions that these enhancers have with the surrounding genes and promoters. We also note that this is a region of open chromatin based on the DNA clusters, and there are also transcription factor binding sites here. We note that there is nascent RNA production occurring here, so there's eRNA being produced at this enhancer cluster. And we also see R loops being formed here. So eRNA R loops are being formed right here. Now, just to point out, there is, in fact, if you look back at our consensus data, there is actually a DRNH only peak right here. And also very interesting, we can see that this is a region that is in between two topologically associated domains of the chromatin. So that is what we call a TAD boundary. Okay, so jumping back to our model here, now that we've pretty well shown that there are, in fact, eRNA R loops being formed at these distal enhancers, can we now understand what types of enhancers these are, what roles and relationships they might be participating in? And so I basically took all of the distal enhancers and iPSCs, and I looked at the types of enhancers they represent. And then I looked at the ones that actually have eRNA R loops forming out them. And we see a very clear enrichment here for the bivalent variety of enhancers. And so I wanted to explore that a little bit further. Just again, showing this enrichment with our MAP-R data, doing a meta-analysis around the bivalent enhancers. We can see all four replicates very clearly enriched within them. So again, eRNA R loops at bivalent enhancers. So before I go any further, I just want to briefly explain what I mean when I'm talking about types of enhancers. So there's the active variety of enhancers, and basically these have activating histone marks such as H3K27 acetylation, and this leads to the transcription of the target gene. We also have repressed enhancers, and these have inactivating marks like H3K27 trimethylation that lead to the target gene being switched off. Now, just a really quick note, you can see here PRC2, that's a polycom repressive complex 2. That includes, for those of you who are familiar, EZH2, and this is what deposits those inactivating marks. Now, I'm going to turn our attention to the poised or bivalent enhancer. These are very interesting. There's something in between active and repressed because they have activating histone marks such as H3K4 monomethylation, but they also have inactivating marks like H3K27 trimethylation. And this is very interesting because this sort of sets up a chromatin environment that is ready to be turned on and ready to turn on the target gene, but it hasn't been activated yet. And so we often see these in pluripotent stem cells like ESCs and iPSCs because they basically prime developmental programs to be switched on in response to a differentiation signal. So you can't tell what I'm alluding to. I then looked next at the genes that interact with the bivalent enhancers that had R loops, and I saw very interesting that they relate to pathways related to the HOX gene families. For those of you who don't know, HOX genes, also known as homeobox genes, are highly evolutionarily conserved gene families that are responsible for large-scale body morphology determination earlier on in development. So seeing this here was quite interesting. Looking at one of these, we're going to turn our attention to the HOX-D cluster here. We can see that it is, in fact, an enhancer cluster based on the presence of open chromatin, CTCF sites, DNase, and transcription factor binding sites. And we can also see that this enhancer is interacting with itself in three-dimensional space based on the high C track here. Okay, so this is in fact an enhancer cluster. We can also see that it has inactivating marks, H3K27 trimethylation. It's also got EZH2 binding, which is what deposits that. It's got activating marks such as H3K4 monomethylation. And we see a lack of H3K27 acetylation. All of these point to this enhancer cluster as being bivalent in character. Okay, now, very interesting. We see high levels of R loops occurring here. And we see RNA Pol II occupancy. And yet, very interesting, we note pretty much no nascent transcription occurring, or at least very low nascent transcription occurring, which is a bit confusing. So I decided to look at some of the other HOX clusters to see if we would find the same pattern. And in fact, I found it at all of them. Here's another example, HOX-A. We see again R loops being formed, and we see RNA Pol II occupancy, and yet pretty much no nascent transcription is occurring. Okay, so I wanted to then look at all the bivalent enhancer clusters and see if that pattern would still hold. And so just to orient us here, these are called tornado plots, and I'm just going to walk through each step so that you can see how they work. This is the meta plot. It's a normal part of tornado plots. And we see that if we look at the bivalent enhancers, they have pretty much no H3K27 acetylation signal, as we would expect, whereas non-bivalent enhancers clearly show an enrichment of H3K27 acetylation at these sites. So this is a heat map showing the bivalent signal, and here's the heat map showing the other enhancers that are not bivalent. I'm just going to highlight this so you can keep track of it. All right, so we jump over to H3K27 trimethylation, which, as we would expect, is enriched within the bivalent enhancers. And we also see H3K4 monomethylation, that's our activating mark, which is also enriched within those bivalent enhancers. We also see here CTCF, EZH2, and open chromatin, ATAC-seq. And as we saw previously, we see both R loops based on our map R signal, and we see RNA Pol II occupancy. And yet again, we also see pretty much no nascent transcription occurring. Okay, so this is all very interesting. And basically what this forced me to do was to update my conception of what's going on at these bivalent enhancers in these stem cells. Essentially, what we can see here from these results is that there is very likely RNA Pol II constitutively paused at these enhancers. And as a result, we see R loops being formed. And based on the last section of the talk, that basically means these are class 1 R loops. Now, the reason for these class 1 R loops is something of a matter of open investigation, open debate. I have a few ideas myself. I unfortunately don't have time to go through them here. If you'd like to ask me about them, feel free to. But that is going to be a very fascinating direction for the future research that results from this study. So jumping into some key takeaways, first of all, I developed a quality control model in coordination with collaborators for R loop mapping data. I found R loop consensus regions covering about 4.32% of the genome. Found support for the class 1, class 2 model of R loop biology, along with finding that drNH can map intergenic R loops at distal enhancers, and that class 1 R loops form across transcriptionally silent bivalent enhancer clusters at developmental genes in these pluripotent stem cells. So some future questions. What is the role of R loops at these bivalent enhancers? What are the other types of R loop roles and classes that exist out there? What is their association with DNA damage? So now bridging the gap between the physiological and the pathological contexts. And then how do R loops change under differing biological conditions? And this is something that I think can be very well addressed by building a database of R loop dynamics and R loop changes in response to various perturbations. Before I move on from this, I'm just going to mention briefly that as a result of this work, there was a lot of software and code developed, and that has now been packaged and provided openly for the R loop biology community. So the first that I want to mention is RLBase, which is a web server and database for the exploration and analysis of R loop mapping data. That was recently published a couple weeks back in Nucleic Acids Research 2023 database edition. And then there's also RL Suite, which is software. It's a software framework for R loop bioinformatics, and that is currently on bioRxiv as of a couple weeks ago. With that, I want to mention just very briefly a couple minor research highlights. This was the first study that I had the opportunity to lead. This was published in Cancers a couple years back. And basically, I used transcriptomic data mining to understand more about the developmental context of Ewing sarcoma, a pediatric bone cancer. This is another study that I had the opportunity to lead, in which I developed a database and web server called Correlation Analyzer that is for measuring gene co-expression correlations and doing a variety of interesting statistical analyses upon them. And these are a few other studies that I had the opportunity to be co-author on during my time as a PhD student. And with that, I want to make a couple acknowledgements. First and foremost, of course, I want to thank the Bishop Laboratory. And I just want to start by thanking Dr. Bishop. Honestly, you've been an incredible mentor to me. I have, really enjoyed the last 4.5 years in your lab, and I'm really going to look back fondly and miss the experiences I've had here. If any of you are new IBMS PhD students, I would highly recommend that you rotate in our lab. I, of course, want to thank the rest of the Bishop Lab. Honestly, it's been an amazing time, and I'm really going to miss your friendship and your camaraderie and your support. I want to, of course, thank my dissertation supervising committee. I have genuinely enjoyed our committee meetings. They've been a blast, and I'm really going to miss them. And of course, I want to thank my funding. And last but not least, I want to thank the Bioinformatics Research Network. It's a nonprofit that, as Dr. Bishop mentioned, I started during my PhD, and many of the trainees who are a part of that research network have been an important part of the studies that I've described today. And so I want to thank the Bioinformatics Research Network for their support. And if you have any questions about it, if you're interested in joining, anything like that, do feel free to reach out. I would be remiss if I did not also thank the friends that I made along the way. We had a lot of fun times. I want to, of course, thank my parents for their support, parents on both sides, especially as I've gone through now 10 extra years of higher education. And I want to thank, of course, my partner, Stephanie. I'm really looking forward to taking the next step in San Diego with you. And of course, our cats. Okay. With that, I just want to briefly mention what I'm doing next. So heading to San Diego, I'm going to be taking a position as a computational biologist at Altos Labs under the direction of Morgan Levine. And we're going to be working on epigenetic clocks and their interpretation of cellular markers of aging and cellular reprogramming. And with that, thank you all so much for your attention today. I'd be happy to take any questions at this time. Yes.
Fantastic. Congratulations. I have a question and a comment. I start by the comment.
Sure.
You mentioned that there is really no positive control for arguments, but I think there's probably one. So in cancer, if I understand correctly, possible to create single-stranded DNA, right? So in the context of tumors, these single-stranded DNA have signatures of mutation by specific mechanism that they need single-stranded DNA, for example, after the transplant, and they have specific mutations in there. So you can probably use that as a proxy. They call it that's the comment. And then the question I have, I didn't see anybody in your talk mentioning anything about the enrichment of these two classes, possibly in endogenous elements.
That's a fantastic question. Just for the people who are on Zoom, there's a comment about how you can potentially use ApoVec as a signature of where R loops are forming in many cancers, as well as a question about, oh, shoot, can you repeat that one more time for me? I'm sorry. Yes. Yeah, I got it. So basically, how are R loops associated and related with these endogenous elements like endogenous retroviruses, line, sign elements, that sort of thing, repetitive elements? That's a really fantastic question. And it's one that we've actually had the opportunity to explore a little bit in some results that I actually did not include today. The challenge, unfortunately, is that the R-loop mapping modalities that we had the opportunity to look at, many of them produce what I would consider to be quite spurious results. And unfortunately, one of the ways that manifests for a couple of these assays is that they're actually enriched within repetitive elements, particularly like ALU repeats, that sort of thing. So it can be a little bit tricky when you're trying to ask, are R loops forming in these elements or not? That being said, when we take our high quality data and we look, we do see very clear enrichment within sign elements in particular, and a little bit in lines as well. But we have to, I think, still explore that a little bit further. It's a great question, though. Any other questions? Sure, go ahead.
So it sounds like as the cell undergoes differentiation, the mapping location to both sides of the cartilage should change. Has that been examined in this department?
That's a fantastic question as well. So basically, she has asked, has anyone looked at where our loop dynamics are occurring, where our loop changes are occurring throughout the differentiation process? Yeah. And yes, they have. However, that is data that has not, the data from that study did not pass our quality filter, unfortunately. They looked at IPSCs, they looked at HESCs, MSCs, BSCs, a few other things, and attempted to see, are we seeing changes in where the R loops are occurring throughout that differentiation arc? But unfortunately, we just really don't feel confident in that data. So we would really want to redo that. Go ahead.
So you're not going to stay and do this, but there are a couple of wonderful differentiation models within adult organisms that could be studied. One of my favorite is spermatogenesis, where you go from stem cell to a fully differentiated sperm animal that would be very facile to that question.
That is a great point. Yeah, well, not me, yeah, but the lab will have to definitely consider that. Yeah. All right. Oh, yeah, go ahead.
So there's been like an association between reflexes and R loops. So do you think that this bivalent, the intensity of bivalency in the enhancer RNA, could that be partly because of G reflexes, or is it kind of like a chicken or egg situation where bivalency occurs after G reflexes or before?
Yeah, that is a great point. So Aola has talked about the association between R loops and G quadruplexes, ostensibly because on the displaced single-strand DNA loop, you have these G quadruplexes that form and help to stabilize the entire triplex structure. Now that's something that we did look at G quadruplexes. So I did look at G quadruplexes, but not specifically at the bivalent enhancers. That is a great idea, though. Yeah, that's definitely something that we should chase up on. Yeah, R loops do tend to be associated with these G quadruplexes, and we can see that in our data. But yeah, no, we haven't looked at the bivalent enhancers yet. Thank you. All right. Well, since we are a little bit over time, should I go ahead and? Okay. Thank you all so much for attending. It's been an honor to get to present this for you all. And yeah, feel free if you've got any additional questions to hit me up over email, or yeah, just come talk to me at some point later on. Okay. Thank you. Thank you all.